and we have no clear evidence what's going on behind this effect, which has been well documented over the years. So there was a big study which included, as a Cochrane review, more than 1,000 patients suffering from low back pain, and they found out that if you apply heat in the therapeutic range means up to 40 degrees Celsius, then there will be an effect which reduces the pain. And this is compared to oral placebo. So they analyzed further if cold therapy as well can relieve pain. However, they didn't find that this cold therapy relieves pain. So there is no, let's say, no clinical evidence in a point of view that there is a sophisticated review of more than 1,000 patients. So what they analyzed further, they thought they could analyze the combination of heat and exercise, and what they found out that if you combine those two treatments, then you get a further reduction of pain. So the evidence for this last point is only 1 or 2 studies, however, it is significant. So this is what a big study says, and so everybody says, or I asked myself, why is this happening? And so basically I want to put you through all the different processes we learned in this course already. And the first point I'm going to address will be the innovation point, and I want to remind you on the data of Professor Menze, who gave the talk on Monday, and he found calcitonin G-related protein in those fascia pieces, and also he did substance P investigations in fascia. Both substances are highly related to pain perception, and you will find them anywhere where you get an inflammation or chronic pain or whatever. And so this is very good data that here in the fascia you will find those spots where you can find those pain substances, basically. So the fascia is really a sensory organ as well. So I don't want to go into detail to this sketch. However, that's just to memorize how he investigated this. And if you inject Freud adjuvants, which is a chemical irritant, into the fascia, you will see that the intact muscle has receptive fields like 5% of the maximum, and then this will increase significantly if you inject this. So the input from fascia will be upregulated in irritation, however this is caused physically or chemically or whatever. So again, this is proof that fascia is a highly sensory organ. However, literature says that chronic back pain is basically, it is caused by ligament, by bone problems, by problems with the facet joints, and so on. The research concentrates on the spine as such. So this is not the truth, as we can see in the dissections of Andrew Fleming, because the spine is surrounded by the erector spinae and the psoas muscle and all the others, which have a fascia around them. So this gives you a lever arm, which is, in my opinion, very important to all the stability of the spine. And you can imagine that in some patients, you will find, in some low back pain patients with low back pain, you will find any problems with your disc. However, there is a big portion where you have low back pain and there is no extrusion. So many people without back pain have disc bulges or protrusions. So there must be something else. And so my hypothesis is that this is probably caused by the fascia. Another study showed that low back pain is associated with diminished proprioception, which again might be down to the innervation of the fascia, and as I showed you, the receptive fields increase, and so the localization will be difficult for the nerves. So if you come down to the temperature effects on the innervation, you need to be careful, because I was really searching the literature. However, there is no clear evidence, because you have temperature effects on the central nervous system. You also have temperature effects on the peripheral nervous system in both directions. Have inhibiting effects, and also you have effects that enhance excitation of the neurons. So the nociceptors, usually they get more excitable if temperature rises. However, there are pain receptors which inhibit the nociceptors. So it is quite a complex topic, and as well there are biochemical pain transmitters. So there is no clear answer to the question, what does temperature to the nociception as such, and the nerval excitation. So the next point I want to address is fluidity. And I want to refer to Dr. Gibnato's work, who did a lot of this. And as you remind, probably the Ulmer Münster, as seen from the university during fog time, that's basically, in my opinion, what we know about fascia. And all the other part below, we have no idea. However, there are great guys, such as the videos of Dr. Gibnato, which you will see later on, that fascia is really an amazing tissue, and you can see how they glide, and this is really a smooth movement. So this is just a very brief sequence of his great videos. Maybe you can answer later on what's your opinion on fluidity and temperature. But as you've seen right now, there are blood vessels running through, and the blood vessels are activating during muscle activity. So if you measure temperature on a limb at rest, this will come down to, let's say, 28 degrees if you really cool it down, and that's different to the core temperature. However, in working muscle, you will heat up the limb nearly to 39 degrees, whatever. If you really work hard and you can feel it, it's simple. If you cool the limb, if you do computer work, whatever, it's much colder than a working muscle. So you can imagine, you've seen all those blood vessels running through, so you can imagine that temperature plays a role as well in the fascia, because this is right where it runs through. So I guess that this fluidity is strongly influenced by the blood vessels and by the temperature inside the fascia. Next point I want to address is the hydration. If you remind Tom Findley's talk, he taught you about the toilet brushes. The toilet brushes are found in several molecules, and this is a proteoglycan, and you can see all those binding sites for water, for hydration. And he set an excellent example that if you jump on several toilet brushes on the floor, you will have a perfect buffer, and that's, in my opinion, how the proteoglycans can work. So that's an excellent example. We did in our lab examinations about the hydration effects, and we had animal fascia, and then we measured the water content in a drying oven, and then we set a zero time. We found the water content in this species, pig species, of 68% roughly. Then we did a stretch of 10 minutes, and then you can see that the water is drained out by the stretch. However, the most interesting part is after this stretch, because as you see, after a while, the water replenishes, and it goes over its initial level, so there is an overshoot. And this obviously depends on the strain rate. So if you have 4% strain, you will get up a little bit. However, if you have 6% strain, this effect is even stronger and it's statistically significant. So what else we did? We have this organ bath, as some of you might have seen during the lab tour, and then you have carbogen to keep the oxygen inside the fluid and to optimize pH level. You've got a surrounding organ bath for controlling the temperature. You've got a highly sensitive force transducer, and then everything is connected to a computer, so you can assess the force inside this organ bath of the specimen in different conditions. So what did we find? So if you look at the force level versus time, the first stretch gives you a curve like this. So you will get an initial fast increase, and then this sample resulted in a force of 40 millinewton, and then fell down to 30 millinewton, and then we let it rest for a while, and then the second stretch shows a tension increase. So in other words, the stiffness has increased, and so we attribute this to this overshoot. If we have more water inside, then the fascia has been soaked, such as the toilet brushes have been soaked, and this is the reason for this effect. We also did experiments by changing the osmolarity in a similar manner, and we could confirm the results. So increased hydration goes along with an increased stiffness in our in vitro experiments. And temperature does enhance the process of taking up water and drainage. And some of you might know this from a clinical point of view. As I talked to Frau Werner, she told me in her experience it is really important to have warm lymph if you do something like lymph drainage. So otherwise you won't be able to get the water out. And so this is another confirmation of my opinion that heat enhances the process of taking up water and accelerating this process. So the next chapter I'm going to talk about is the myofascial tone. And so in this myofascial tone you will learn about the tonicity of fascia and on the other hand the tonicity of muscle, because it's always a balance. You have in your tissue, I don't know if everybody can see this from behind, but this is a piece of muscle and you can see the white bit inside, and this white bit, as you can see histologically, is a piece of fascia basically, or in other words, perimysium, endomysium, and how you would call it inside. But it's connective tissue which is connected and which is surrounding and the muscle. So if you look at these pictures, if you look at phasic and tonic muscle, the amount of connective tissue inside will be different. Phasic muscle contains less thick bundles of connective tissue, whereas tonic muscle has thicker fascia pieces, so probably the fascia takes over more load-bearing function in those tonic muscles. So we are talking about the temperature effects. So what does temperature on this? So in my opinion, you need to first look at muscle and then look at fascia. What does temperature do those things? If you remember physiology books maybe, then this is a sketch of a skeletal muscle cell which has a big calcium store inside, and then the contractile filaments. So the extracellular space will be activated via acetylcholine on this motor neuron end plate, and then you will get activation, depolarization, so the action potential is coming over to the muscle cell, and this will in turn run along the T-tubule, which is deep inside the muscle cell, and then through the biggest receptor our body knows, or we can see it even under the light microscope. It's so important because it releases calcium very fast, and you can play piano with this because it's really fast, and then taken up again, there is the contraction. So that's how muscle works. And you can see this is a lot of different biochemical pathways going along. So basically, if you look at calcium in what happens if you put here a potassium signal, then you will see the sudden rise of calcium. So it's really, really basic what you see in muscle tissue. So maybe again, you can see that the green line is an indicator for calcium. Yes, you can even see it moving. This is a dissected muscle fiber under the light microscope, and this is the corresponding calcium imaging. So now it's moving, and the calcium is, by the way, this was found in Ulm, that calcium activates the muscle tissue, and this is the source of contraction. So you're at the part of, and it's worldwide accepted now. So if you look at different temperatures, you can see that at 20 degrees Celsius you will get a twitch, a muscle twitch, which is a bit slower and not so high. Then as you warm up the muscle, as you go along the temperature, you can see that the peak of working muscle is really sharp, and the relaxation is faster as well. If you do a statistical plot, so statistical analysis of this, and you plot the temperature on the x-axis and the relaxation time and the time to peak, you will exactly get, please note that this is a logarithmic scale. You will exactly get the logarithmic law of all biochemical reactions. So basically, what working muscle does is a biochemical machinery, and you got it activated by temperature. What is the case in fascia? Let's have a look at those charts. We did basically a similar investigation, and the blue line represents the cold fascia, the red line represents the warmed up fascia, and you can see that the blue line has higher forces than the red line. So in other words, temperature leads to a facial relaxation. So we confirmed basically all those other guys back to the 50s who found this in their experiments in another setup, whatever. However, this is a more recent one of Dr. Huang, which was published 2009. Basically, he did similar experiments, same stuff, 70 degrees, 27 degrees, 37 degrees. He had the same results. So he also did this logarithmic plot, this temperature plot, and he found this temperature dependence here, which is not logarithmic. So it's the other way around, as you might remember, the muscle curve. So if I want to summarize the temperature effects on fascia, in high temperature the stiffness is lower and the peak force will be lower in terms of fascia. Remember, muscle will have a higher peak force. So, and in low temperatures the stiffness will be higher and peak force will be higher. So this is conflicting. If you remember all this this stuff I told you about muscle tissue and the in vitro data on fascial tissue, this seems conflicting. However, it's woven together, so you would never tear it apart. You would never say, okay, I just look at the muscle, I just look at the fascia. It's one entity, and so I think if you combine this, it is from a physiological point of view, it is very, very great what the body did, because if you are at rest, the fascia are stiffer because they are colder, so they prevent you from, they have a higher load-bearing function in cold surrounding. And as you work and you go along, or you bear a log on your shoulders, whatever, muscles heat up, and the range of motion will increase, and the stiffness will decrease, so you have a higher range of motion, and you have a better possibility of exercise. So this truly makes sense, and this is a very good combination of those two on the first hand conflicting results. So last but not least, I want to just have a brief view on the contractures. And you know this from Robert Schleib's talk that in myofibroblasts have been found in many fascial contractures, the Pilgrim, Leatherhouse, frozen shoulder, and many others. In all those diseases, you will find fascial shrinkage and the involvement of myofibroblasts. So this is a short video of one of those guys going along inside the fascia, and this is really amazing how this fibroblasts and myofibroblasts can go through the fascia. So Robert analyzed several pieces of fascia, and he could find differences in different locations, how many myofibroblasts there were. So for instance, at the lumbar level L2, there were significantly more myofibroblasts than at the fascia lata, for instance. So this is a stain of alpha smooth muscle actin, and you can see all the red spots which represent contractile filaments. So contractile filaments indicate the presence of myofibroblasts. Or in the small inset here, you can see the digital quantifications. That is the method how it was done. So Helen Larchevo, you will hear her talk this evening. She found that lumbar fascia is significantly thicker in low back pain patients. So maybe this has to do with the involvement of myofibroblasts and this remodeling I've showed you in this little video. So again, we did our in vitro studies, and we found that that the myofibroblasts, they do not respond to transmitters of the autonomous nervous system. So this was a surprising result for us. And they do not respond to general muscle activators, most notably caffeine, which is a very, very strong activator of muscle in vitro. So Robert was checking several other stuff, and he found mepyramine, which gave a strong response in fascia. Where it is, so you can even measure it is in several Newton, going up and going down. So it has an effect on the force. So how could it be, on a biochemical point of view, that those muscle activators do not work, even those for smooth muscle do not work? Because you have probably both pathways inside the myofibroblasts, one which is calcium sensitive, which leads to a fast contraction in smooth muscle cells, and there is another pathway which leads to sustained contractions, and this is calcium independent. So all the stuff I showed you about calcium is about phasic contractions. And if you look at this pathway on the right hand side, it is for long-term contractions and long-term load-bearing functions. So probably, or we could prove almost, that myofibroblasts only have this right-hand side pathway. Again, in terms of the lumbar fascia, we found there are myofibroblasts involved. We found that there is a lever arm here, and we found that the myofibroblasts have a contribution to the force generation in the fascia. However, there is no study which investigates the temperature effects in vitro on those myofibroblasts. So this has to come in future, has not been evaluated. But if you look at this guy going through fascia, I can imagine that the remodeling is strongly dependent on temperature as well, and that you will get temperature effects on proliferation of myofibroblasts. Because usually in human beings, temperature enhances growth, cell growth. So just coming back to the initial hypothesis of this just pain generation just in the spine itself, I want to introduce the hypothesis of frozen lumbars. If you have frozen shoulder with invasion of myofibroblasts, you could have in low back pain the frozen lumbars as well. And if you keep on training, that's what the Cochrane review said, then you probably increase your temperature, and besides all the other effects I told you, at least you can have definitely a heat-induced relaxation, and this might be worth to stop this vicious cycle of no activity, no perfusion, temperature goes down, and then you even get more contraction, more pain, and this is a vicious cycle where I think heat is a partial way of reversing this vicious cycle. So I also showed you the effects of innervation, where there was until now no clear answer, and I think the topic is too complex that you can answer it, the temperature effects on this pain tract. And I showed you the fluidity, which will be addressed by Dr. Klingler. And I also want to show the video of Archon again, because if you look at these ladies, how they walk, they are really swinging with their lumbars, and I can imagine those lumbars, if you measure temperature inside, they are much warmer than probably in myself when I walk without this swinging lumbar walk. So I think this is a very good example of keeping the muscles warm. So last but not least, I want to address the limitations. So no heat therapy in acute inflammation or skin lesion, or very important in peripheral neuropathy, because people, they won't be able to sense the heat, and so you can have the risk of burns. And also in other diseases like thalamic ischemia, the sensory organ is not working properly, so you have the risk of burns. So just for stopping the talk.